So the next one that we are going to see is biomacromolecules. So biomacromolecules means the molecules which are generally large in size with higher molecular weight. So higher molecular weight means it will be in terms of Daltons. So mostly it is greater than 10,000 Daltons. Okay, so all these biomacromolecules which are there, they are generally called as what? Polymers. So poly means many. Okay, so polymers are nothing but chain of monomers. So when these monomers link together, they form this biomacromolecules, which are generally known as what? Polymers. Okay, so in this, we'll be studying about the proteins, then certain polysaccharides, in nucleic acid and lipids. Okay, but out of this four, lipids all are not macromolecules. Okay, some also come under the micromolecule as well. So the first one we'll see is the protein. So protein are nothing but polypeptides. Poly means many, peptides means now proteins are made up of the peptides of amino acids. Okay, so when they join together, we get something which is called as polypeptides, and basically they are what? Proteins. So in short, proteins are what? A long chain of amino acid. Chain of amino acid, which are joined by the bond which is called as peptide bond. Okay, so mostly each macromolecule is having a type of bond, okay, which links them together. For example, now protein we already know it's what? For the protein, the bond name is peptide. If you take lipid, in lipid you have what? Ester bonds or ester linkages. Then if you take carbohydrates, means the sugar units. In that the bond name will be what? Glycosidic bond. Then nucleic acid. Okay, so nucleic acid means that nucleotide, that is the sugar base, phosphate group, right? All this nitrogenous bases which are there. So they have the bond which is called as phosphodiester bond. So these are what? Important for molecules and their respective bonds. Okay, so this is very important, which type of bonds are present in this macromolecules. So coming back to the proteins. So proteins are nothing but what? The long chain of amino acids which are joined together by the peptide bonds. So it is not that the polypeptide chains will be same. So it may be different. So you can also write that proteins are mostly what? Heteropolymer. Of amino acids. So same amino acid joining only you will not see. So they will be hetero, means variations will happen in them. Okay, so if you take a particular polypeptide chain, for example, we saw that in a polypeptide chain, so amino acids are present, right? So amino acids like glycine, leucine, so on. Fine. So here there will be what? A bond over here like this, leucine, alanine. I'm just randomly writing this. Okay, asparagine like that. So in this chain, okay, in this polypeptide chain, you have basically two terminals of the amino acid, out of which the first amino acid terminal is called as what? N-terminal. And the last one is called as the C-terminal. Okay, that is OH and NH2. That is C-terminal and N-terminal. So the first, whatever the polypeptide chain, the first terminal is there, that's N. Whereas the last one, it is called as what? The C-terminal. Okay, so you have many kind of proteins which are generally present or which are externally taken. And some of the examples of proteins are what? Collagen. Okay, basically which is present in your skin or in animals it's present, right? Then you have certain hormones or enzymes. Hormones, if you can take, is insulin. Okay, now insulin is a hormone for the sugar patients they are given. So this is also a protein. Then in enzyme, you can take trypsin. So there's a saying like all enzymes are protein, but all proteins are not enzymes. Okay. Then one more thing is which is generally which are made is the antibodies for fighting against infections. So these are what some of the examples of the protein. Okay, so as protein is a biomacromolecule, so obviously we'll be having a complicated or complex structures. So let's see the structures or structural level of the proteins. Okay, so structural level means four are there. Okay, four structural levels are present in the protein, which are generally termed as what? Primary, secondary, tertiary, and quaternary. Okay, so first let's see the primary structure. So primary structure means it will be a simple structure. There will be no more complexity, means bonds will be very easy to break. Means if a protein is in primary structure, you can break them easily. Okay, so here, whatever the protein is there, in that just one amino acid will be joined to the another amino acid like in a simple way. Okay, so you can just take a line. So if you take a line, it's just simple. Okay, you can just break it down easily. So that in that line, again, as I told you, two terminals will be present. That is the N and C respectively, right? So this protein, okay, it is not like very hard. So you can easily break down. So the feature of this is what? Easily destructed or denatured, you can say. So if this has to become more harder or it shouldn't be denatured easily, what should happen to it is it should become more thick or complex structure it should become into. So we have what? The secondary structure. So secondary means it will fold. Whatever the single line of protein is there, it is going to fold itself. Okay. Like for example, it may be like this. And this is termed as helix or like this. That is beta pleated sheet. So in secondary structure of the protein, okay, there are two types mainly, that is alpha helix and beta pleated sheath. So the very general term helical helix means what? Helical structure there will be, and the sheeted means in sheet structure, pleated like sheets it will appear into. So whatever the bond by which this helical structure or beta pleated sheath is stabilized, it is nothing but the H2 bond, that is hydrogen. Okay, so now again when the further, what do you say, folding happens, it turns into the third structure, which is the tertiary structure. Now tertiary structure will be very long chain of protein because it will be folding again and again. So it'll be like somewhat like a woolen ball, okay, or a thread ball. So like that it'll appear hollow woolen ball. So it's a long protein which appears as hollow woolen ball. So 3D structure. Tertiary means what? 3D structure. So here, like as we saw in the primary, most probably bonds will not be there. That's why easily the protein denatures. In the secondary, we saw the bonds which stabilizes these two structures as helical and sheeted. They are hydrogen bonds. Now, when it comes to the tertiary, because the complexity increases, over here the bonds which are involved is also more. Okay, so the bonds involved in the tertiary structure is what? First one is the hydrogen. Second is ionic bonds. Okay, then van der Waal interactions. Then you have the covalent bond and the hydrophobic bond. So when the protein is in tertiary structure, it will be more difficult for it to disintegrate or denature. Okay, that is why in this tertiary structure, your protein molecule gets what? A 3D structure. Then the last one is what? Quaternary. So quaternary structure means whatever polypeptide chains are there, they will rearrange themselves and they will refold themselves. Okay, so here what happens is the individuals which are already present, they will again fold into the polypeptides. Okay, so this thing is called as what? Quaternary structure. Okay, so the simple four structures of the protein. Now, this was on the basis of what, how the structure is of the protein. Now, how does, what is the composition of that particular protein? On that also, two types are there. So, classification of proteins on the basis of composition. So, they are generally of two types. Okay, that is fibrous and globular. So fibrous means what? Whatever the protein chains are there, they will be parallel like this. Okay, and they are held together by two bonds, that is H2 and sulfide bonds. For example, you can take here is the collagen or the keratin which are present in nails and hair, fibrion, myosin. Okay, all this stuff. These are what? The examples of fibrous means whatever the protein polypeptides are there, that chains will be parallel to each other and they're held together by two bonds, hydrogen and disulfide bond. Then second one is the globular protein. So globular means globule like it will be. Means it will be what? Coiled. Okay, or you can say like a sphere it will be. So spherical. And some of the examples which we can take over here is the insulin, hemoglobin. All this will be what? Globular protein. Okay, so these are the two types on the basis of composition. Now, there are again some types, okay, depending upon what, how they are derived or like if they are simple or they are complex structures or complex protein. So the B one is what? On the basis of molecular component. So in this three types. So the first one is the simplest form, so simple proteins. Then you have conjugated proteins and derived protein. So in the name itself, it explains the protein. Okay, simple protein means what? Only amino acids will be present. Means only protein part will be present in them. Okay, so you can write it as made up of amino acid only. So examples, collagen, albumin, which is present in egg. Okay, these are some of the examples. In conjugated, now in simple proteins, only proteins will be present. But if you take the conjugated proteins, they will be made up of what? Protein plus non-protein part. Protein plus non-protein part. For example, hemoglobin, okay, which is present in blood, or casein. This is present in milk. Then comes the derived protein. So derived protein means whenever a natural protein breaks down partially and forms another molecule, then that is called as what? Derived protein. Okay, so this is formed by partial breakdown of natural protein. So here the examples will be insulin, fibrin, peptones. Peptones are also forms of proteins only. Small proteins are called as peptones. Okay, so these are what? The two types of classification, that's on the basis of composition and what molecule components they contain. So the next biomacromolecule, that is the polysaccharide. Saccharides means sugar, poly means many. So we are going to see many sugar components over here, which are generally termed as polysaccharides. So what are polysaccharides? They are nothing but the polymers which contains minimum of 10 monosaccharide units. Okay, so we already saw the like the monosaccharide, okay, the polysaccharide, oligosaccharide. So monosaccharide means only single unit will be present. Oligo means 8 to 10 units of the sugar, and poly means more than 10. So minimum you'll be having what? 10 saccharide units. Okay, so this particular polysaccharides, they are being divided into two types. Okay, so the first is the homopolysaccharide. So homopolysaccharide means what? Homo means same. So the second type will be what? Heteropolysaccharide. So in homopolysaccharide, whatever the sugar units are there, they will be same. Okay, like for example, take a sugar glucose. So that entire chain will be composed of what? Glucose only. Okay, so homopolysaccharide, or the alternative name is homoglycans. Okay, so they have what? Only one type of monosaccharide unit. Okay, so if you take the chain of glucose, we have the examples such as what? Starch, or you can take cellulose or chitin. So this contains what? A chain of glucose units only. Fine. Then the second type, which is what? The heteropolysaccharide. Also called as heteroglycans. So here it will be what? Instead of a similar kind of monosaccharide unit, you will be having at least two types of monosaccharide unit. At least, okay, so there can be many also. Two types of monosaccharide units. For example, pectin or peptidoglycan. Okay, now pectin is something which you will find in fruit. Okay, the fruit skin, whatever is there you will find the pectin. Okay, whereas peptidoglycan means mostly in the bacterial cell wall. You will find this. Okay, so as I told you some examples, like out of these examples, we'll just see a glimpse of the polysaccharide examples. Okay, so some polysaccharides For example, the first important thing is the starch. Basically, starch where it is present. In plant, starch is present. So the storage polysaccharide of a plant is nothing but the starch. So because it's glucose unit chain, so what will be the formula? C6H10 O5N. Okay, so this is what a polymer of D-glucopyranose. Okay, so two types of rings are there when you take a structure of any molecule. Two types of rings will be there, pyranose and furanose. Okay, so pyranose means six-membered ring like this, whereas furanose means five-membered ring. So basically, in the glucose you have this pyranose, and furanose you have in the fructose. Okay, glucose, pyranose structure, fructose, furanose structure. That is five-ring structure. Okay, so starch is what? A polymer which is made up of D-glucopyranose. So here there will be a linkage for every molecule. So here the linkage is what? Alpha 1,4 glycosidic linkage. As I already told you that in the sugars, the linkage is what? Glycosidic linkage. So the link between the first and the fourth. So alpha 1,4 glycosidic linkage. So starch is a typical complex molecule because it doesn't easily get dissolved in water because of the two components which is present in the starch called as amylose and amylopectin. Okay, so amylose is basically a simple structure. Okay, so but here branching comes in the amylopectin. That is why the starch only on heating with water only, it will form a gel-like structure. Otherwise, it will not dissolve in water. Okay, it is because of these two components, that is amylose and amylopectin. Then the second one, which is mostly present as a storage polysaccharide of animals, which is glycogen. Okay, so here this is also made up of what? Glucose unit itself. So in plants, the storage polysaccharide is what? Starch. Whereas in the animals, the storage polysaccharide is the glycogen. Then we have the next one, which is cellulose. So cellulose is a polymer, okay, which has what? A linkage of beta 1,4 glycosidic linkage. So here also it's a glucose molecule only, but it will be a beta form. Beta D-glucose chain will be there. Then you have the last one, which is the chitin. Okay, so chitin, basically this is also a linear polysaccharide only, but here it is unbranched. Unbranched heteropolysaccharide. And the linkage will be beta 1,4 glycosidic. So this is actually in fungal wall, you will find this thing, chitin. Because chitin is a this one, the polysaccharide which is present in the cell wall of what? The fungus. Okay, so these are what? Some general examples of the polysaccharide. Then the next type of biomacromolecule, that is nucleic acid. So already we know about nucleoside and nucleotide. Okay, so nucleic acids are nothing but what? The polymeric components of nucleotides. So this is also basically what? A polynucleotide. Okay, so if you say a nucleic acid, already I've told you that in the like viruses, the genetic material is different. Okay, in the humans, and animals, the genetic material is different. So we have two things. Okay, one which is called as the DNA. The full form is deoxyribonucleic acid, and then you have the RNA, which is ribonucleic acid. Okay, so what is this deoxyribo means the DNA as well as the RNA. So first, let's see about the DNA. Okay, so DNA is a, what do you say, a helical structure. Okay, a double right-handed helical structure of two parallel polynucleotide chains. Okay, so somewhat it will be like this. Okay, so you will be having a 3 prime, 5 prime, and here will be 5 prime, 3 prime. So these are basically anti-parallel chains. So see, anti-parallel means what? Here you can see 3 prime to 5 prime, but the other chain is what? 5 to 3. So they are what? Anti-parallel, then double helix you have already, and they are right-handed helical structures. Okay, polynucleotide chain. So this structure which is there, right-handed double helical model, it was given by Watson and Crick. Okay, which also has what? Certain grooves. So this grooves is in a particular angle. Okay, and these are joined together by what? Already I told you the nucleic acids have a phosphodiester bond. So over here, phosphodiester bond will be present. And here you will be having AT, GC. Okay, like that, bonds will be there. So AT means double bond and GC means triple bond. Right? So in one twist, you will find 10 base pairs. Okay, like this 10 base pairs you will find. So anti-clockwise, whatever the twisting is there, that happens over here. So if you want to see write about the feature of the DNA, simple is what? First of all, it consists of two helically twisted strands, which have what? Inside them, what is present over here is the base pairs. So this base pairs will or this chain will be aligned in what? Anti-parallel or opposite directions. So each of the helical means this helix or the round which is there, that is around what? 20 angstrom. Okay, and one turn, if it is completing over here, it is 34 angstrom. Plus it has what? 10 base pair. So these are what? The first feature of the DNA strand. Second one is what? Whatever these two strands are there, okay, when they are in the clockwise direction, what is the form of them? A right-handed helical. But the arrangement which they have is the anti-parallel. Okay, whatever the twisting is there, that is what? Clockwise twisting with the strands which are arranged how? In the anti-parallel form. So if you see each of the strands, they will be having a backbone. And that backbone is nothing but made up of what? Two things. That is a deoxyribose sugar and a phosphate molecule. So when this phosphate is going to join the sugar, then the bond which is formed is called as what? Phosphodiester bond. Okay, so because of this bond only, whatever the DNA is there, they are stabilized. So whatever nitrogenous bases are there, that is 10 base pairs, they will be present where? Inside this helix. Okay, and I told you this base pairs which are normally known as A, T and GC, that is the pairing between them. They are paired with what? Hydrogen bonds. So between A and T, there are 2 hydrogen bonds. Between G and C, there are 3 hydrogen bonds. Okay, so these are what the features of DNA. Then comes the second one, which is the RNA. So like the DNA, there is no such, what do you say, complexity over here. So it's just a single, what do you say, structure, single helical structure it will be. So this is mostly you will find where? In the viruses. and the name is what? Ribose nucleic acid. So ribose sugar again. Okay, so there DNA means deoxyribose sugar was present. Here it is what? Ribose sugar will be present. So basically you will find this in the single strand. There will be no two strands over here. Okay, so in the retroviruses or the coronaviruses, there you will find this RNA. So there are basically three types of RNA. Okay, that is the rRNA, mRNA, tRNA. So rRNA means what? Ribosomal. mRNA means messenger. And tRNA means transfer. These are what? The three types of RNA. So only if mRNA is there, then only what happened? Protein synthesis will happen. Okay, so mRNA is very important over here. Fine. Then you also whatever the transfer RNA is there, it is also called as what? Soluble RNA. Okay, so these are what? The features of the ribonucleic acid or the RNA. Okay, so the most important thing in all this is what? The bonds. That is very important. Okay, because whenever a monomer is being converted into a polymer, that is when many monomer comes together and forms a polymer, the bonds are the thing which gives what? Its stability. Okay, so if you take any of the bond, for example, if I take peptide bond, where they are present, that you should know. Okay, so they will be present where? In proteins. So how these bonds are formed? When the carboxyl group of one amino acid reacts with NH2 group of next amino acid, and when they are doing this, there will be what? An elimination of water molecule. This is how a peptide bond is formed. If we take a glycosidic bond, that is carbohydrates. So in this, what happens is whatever the ions of the carbon is there, they will just go and bind to the sugar units. Okay, and here also there will be what? A water removal process. In the same way, if we take the phosphodiester bond, which is present in the nucleic acid, just now we saw. So in the nucleic acid, how this phosphodiester bond is formed is already I told you over here. Okay, that is deoxyribose sugar and the phosphate together will give you what? A phosphodiester bond. Means whatever the 3' carbon of one sugar of one nucleotide will link to the 5' carbon of the sugar of the next one, that is the succeeding nucleotide. Okay, so how the bonds are formed? The bond is basically formed between the phosphate and the hydroxyl group, OH group. Of whom? The sugar. Okay, and that bond is called as what? Ester bond. So when there are ester bonds on two sides, then the name as it suggests, phosphodiester. Ester bond. So these is how the three types of bonds are formed. That is peptide, glycosidic and phosphodiester bonds.